Okay, so this lecture is about stem cells and therapeutic cloning. So first let's talk about what is a stem cell, or what makes a cell designated as a stem cell. And it has to have a few properties. So stem cells can continuously generate new cells. So the stem cell here can continuously make new cells that will then go on to differentiate and become different tissue types. One of the main reasons is because telomerase is on in these cells. And hopefully you've heard about telomerase. Telomerase is the enzyme that makes the ends of your chromosomes. So your telomeres are at the end of your chromosome. And what happens is every time a cell divides and replicates itself, because of the way DNA replication works, you lose a little bit of your telomere. So you lose a little bit of your chromosome every time a cell divides. And because in most cells, telomerase is off. So the telomeres get shorter and shorter to a point where they're short enough that the loss of more genetic information could possibly lead to mutations. So telomeres are non-coding. So they don't encode for any proteins. They're just a repetitive sequence, kind of like a buffer. So when that buffer gets too short, the cell actually undergoes apoptosis, which is programmed cell death. And because it wants to avoid gaining mutations or loss of DNA that would cause the cell to act abnormally. So because telomerase is on in the stem cells, they can continuously make new cells because they don't have to worry about the loss of the ends. So the telomerase will continually keep the ends of the chromosomes at the right length. Another important aspect of a stem cell is that it can differentiate into different cell types. So think of this as, I think of stem cells as like the baby cells, and they haven't figured out what they want to be when they grow up. And once they figure out what they want to be, they get those skills. So they turn on those genes, and that's called cell differentiation. And once you become a certain cell type, you can't go back, right? So once you're red blood cells or neuron or adipose fat cells, you can't change to another type of cell. You've already picked your destination. So stem cells are back here that continuously generate new cells and can differentiate into different cell types. Now, how much it can differentiate depends on the type of stem cell. So there are three main levels of stem cells. Totipotent is up here, and you can see that these are cells of an embryo between 1 and 3 days. So totipotent stem cells can become anything. They can become lung, pancreas, heart, neuron, any kind of cell. And we really can't work with these cells because they are produced and then differentiate so quickly. We don't have a mechanism of harvesting and working with totipotent embryonic stem cells. We will talk a lot about pluripotent. Pluripotent, all they have become is one of the three main tissue lines, so endoderm, mesoderm, ectoderm. So you can get endoderm pluripotent embryonic stem cells that can differentiate into all the different types of endoderm tissue. Likewise with the ectoderm or the mesoderm. So pluripotent is embryonic stem cells are pluripotent. I guess I should say it that way. We'll talk about how we get those, but they can differentiate into the most because we just don't have the ability to work with totipotent. Multipotent, also sometimes called adult stem cells, they've already started to differentiate, but there's still, some of them can still become different stem cells. So like your bone marrow can still differentiate into different types of white blood cells, red blood cells. So your bone marrow is a type of multipotent stem cell. It cannot, I should put it, it's over here. It cannot become skin or neuron, so it's not the ectoderm line, but it can become a lot of different types of blood cells. Whoops. Okay. And we'll talk a little bit about where we get all these. So this is just an overview slide. Again, trying to kind of show you, we have tissue stem cells over here, which are your adult stem cells. As it says, many tissues have these types of stem cells to renew. Embryonic are the pluripotent. These are multipotent. And induced pluripotent stem cells we'll talk about is where you take an adult cell and kind of make it go back to being an embryonic stem cell. So let's talk about these different. So first of all, where do you find embryonic stem cells? Well, from an embryo. So again, these are pluripotent. You're harvesting them from a blastocyst, and a blastocyst is generated at about 1 to 2 weeks of cell division. So here's your fertilized egg. It's starting to differentiate into an embryo, and then at this blastocyst we can get these different stem cells. And you have about 150 to 200 cells here. One of the common ways that this is done is through in vitro fertilization. And this sounds bad, but leftovers, meaning couples have donated egg and sperm, they've been fertilized, and they'll grow up these little embryos in the petri dish, and then they'll implant some of them, and some of them they'll freeze down for later. And so if the people do not want to implant any more embryos, you can actually use these embryos to make embryonic stem cells. This has a lot of ethical disputes along with it. Some people don't feel this is right, since this could potentially have been a child, even though we're not implanting these cells. So private companies can still do this kind of embryonic stem cell work. I believe government agencies cannot. It's kind of tied to the abortion issue. But there are embryonic stem cell lines, so tissue culture, we've talked a little bit about, that can be used for government-funded research. It's just like with any cell line, it gets to be a more artificial, and it's the same genetics used over and over. So that that is where you can get embryonic stem cells. Where do you find adult stem cells? So these are also called, you can see, somatic. So remember, you have your germline cells, which are your reproductive cells, and then all your other tissue is called somatic. So there are somatic or adult stem cells that you can find all over. I always thought a cool one was in your teeth. So I used to tell my kids that when they lost a tooth, I would take their stem cells and I could clone them. I'm a weird mom. Like, why do you tell young kids that? But anyways, I thought it was kind of cool that teeth had stem cells in them. Excuse me while I drink my coffee this morning. So, lots of locations. These are all multipotent. So they've all differentiated somewhat. The challenge with adult stem cells is that there's very low in numbers. And they're a lot of times hard to isolate, and they're difficult to grow in the lab. So they're very fragile cells. You've probably heard of bone marrow transplants. That's kind of the most common place we can get stem cells from. And I've heard it's quite painful. They stick a big needle through your hip somewhere to get into the bone marrow and suck out those cells. But there are other places. This doesn't have it, but even your lungs have some adult stem cells. So we're trying to figure out ways to harvest these stem cells for therapeutic uses, which we'll talk about at the end of this talk. But I just wanted to give you a little bit of idea of where you can find adult stem cells, but also what the challenges with them are. So I keep emphasizing pluripotent, which is less differentiated than the multipotent. As cells mature, so as they go from a stem cell to, say, intestinal cells or muscle cells or nerve cells, they start to differentiate and they lose their ability to diversify, to become all these different cells because of epigenetics. So epigenetics is information literally on your DNA or on your chromosomes that controls gene expression. And a lot of these are more permanent mechanisms of controlling gene expression, and so it's hard to reverse epigenetics. So pluripotent has the most potential that we can isolate, and so we want to get these pluripotent stem cells. But as you've seen, the source of pluripotent stem cells is embryonic tissue, right? And so I mentioned embryonic tissue, one, is somewhat controversial of how it's generated from an embryo. But also another thing is embryonic stem cells, if you're using them for therapy in an adult, they have different DNA. So I'm sure you've heard of liver transplants or lung transplants or heart transplants, where you're taking tissue from one person and putting it into another. And the challenge with all those transplants is that the patient has to be on immunosuppressive drugs because they don't want the body to reject the new tissue. So that's also an issue if you use stem cells from a different source. So embryonic stem cells are going to have different DNA than your stem cells. So how could we get pluripotent stem cells from an adult? Okay. And that's called induced pluripotent stem cells, or I with a little I, PSC. Um, Induced pluripotent stem cells are like turning back the clock. Your own DNA, and we can make these stem cells differentiate into tissue that you might need for replacement tissue, let's say. Just want to also mention one other source of embryonic stem cells is umbilical, if I spelled that right, cord blood. So if you had the forethought to harvest umbilical cord blood from your newborn and then store it later on, if your son or daughter had some kind of issue that needed tissue replacement, you may be able to go back to that umbilical cord blood, harvest their own embryonic stem cells, and do therapy. I would guess most of you don't have umbilical cord blood stored away. I did not do that for my children. It was just kind of coming out back when they were born, you know, and it's just kind of also one of those cost things. It costs to get it harvested, and then you have to pay yearly to have them stored for you. It's not a bad thing. It's just probably something that most of us don't have. So we would have to go the induced pluripotent stem cell route. So how do we make induced pluripotent stem cells? So you're taking adult cells. So fibroblast cells, sometimes they use skin cells, sometimes they use blood cells, but fibroblast is the most common. And if you remember, fibroblast cells produce lots of your extracellular matrix in your body. So we can take those cells, there's plenty of them, easy to harvest, and we reprogram them. So there's a series of transcription factors that are added to the adult cells, and they reprogram them by removing a lot of the epigenetic signaling, or epigenetic marks. So epigenetic marks can be DNA methylation, they can be histone acetylation, histone methylation. And so these transcription factors can help remove these epigenetic marks, reprogram the cells into stem cells. Once you have these induced pluripotent stem cells, now you can make all different kinds of tissue. This is a pretty good video. If you have time, watch that. This sounds really great, but it does have some challenges. One of the biggest challenges is the low efficiency. I don't know if I spelled that right, but last time I read, it was about 0.02% efficient, right? So you've got to harvest a lot of cells, do this reprogramming with a lot of cells to get just a few induced pluripotent stem cells. The other big challenge is cancer. Okay. So you are reprogramming these cells to become a highly proliferative, or a cell line that can reproduce and change into lots of different types of cells. Some of the transcription factors, like c-Myc, well, c-Myc is a proto-oncogene. So what they've found is that in this method to induce pluripotent stem cells, they end up with a lot of cells that are dividing out of control. That's basically the definition of cancer. So there's a lot of challenges with low efficiency, and you also don't want to give somebody cancer to repair their tissue. This next slide is just kind of showing you a little bit of history of how do you reprogram, right? So how do you get these transcription factors into the cells. And what it started out was using viruses, right? This would be an ex vivo virus challenge, so, or not challenge, treatment, right? So you're taking the cells out of the patient, adding virus that will express these different tissues. The challenge is that these viruses, retroviruses, like to integrate. And we talked about in gene therapy, if you integrate DNA into the wrong spot, you can also cause disease or cancer. So they used transposons, they used different systems to try to get integrative. Then they said, maybe that's not so good, so let's try non-integrative. So adenovirus is a virus that doesn't integrate. Plasmids don't integrate. Minicircle and episome, so like basically again putting a different type of plasmid into the cell to deliver these genes. You can see what happens is your efficiency goes way down. Next generation, where they're having the most luck, is using actually RNA. So let me just show you. So you can see it's got much higher efficiency than these non-integrative. RNA is not going to integrate into your genome. And it's going to be temporary, so it's going to produce the proteins needed and then kind of shut off because RNA gets destroyed in the cell. So you can go, I put links where you can find more information if you want, but this is self-replicating polycistronic RNA. Polycistronic means multiple genes in one RNA. So when the ribosome comes to translate, it can translate this gene and this gene and this gene and all of these genes in a row. So you put this RNA in, and the cool thing is it's only one thing you have to transfect. So the RNA contains everything. So you're not trying to get 8 or 9 different things into the cell at one time. The RNA is, excuse me, produced, your proteins are produced, and then this technology it somehow promotes degradation after the RNA has been expressed. So it's transient or temporary expression. Right, of the genes. And then once the reprogramming has gone on, you've got now human induced pluripotent stem cells to use for your therapy. So, not perfect still, still is something that takes a lot of research and money, but we are coming a long ways into being able to make these induced pluripotent stem cells. Another way you could create your own embryonic stem cells is something called somatic cell nuclear transfer. So what you're doing is you're taking the nucleus of a somatic cell, could be a skin cell. So it's your nucleus, and you're putting it into an egg that has had its nucleus removed. So think about this. Eggs are normally haploid. They need a sperm to become diploid. What we're doing is we're reprogramming this egg with your diploid nucleus. So that egg doesn't need fertilization. It's just your DNA. And then we can grow it up into embryonic stem cells. Just like I showed. The issue with this is it requires an egg donor. What does that mean? Well, one, that means you have to get females willing to donate eggs. And if you're a male or a female that is not using an egg from themselves, you're going to have different mitochondrial DNA, which really shouldn't be a problem. Just know that you'd have somebody else's DNA in this new tissue in the mitochondria. But like I said, you have to get people to donate eggs, so this is very expensive. So I guess that's all I want to say about that. And I could not find what the efficiency for this is, but I'm guessing it's still not super efficient, just like in vitro fertilization is hit and miss. So it's kind of a similar idea. So why would we want to make our own stem cells? Well, the idea is a concept called therapeutic cloning. So you're using your DNA to make new tissue. So if you're doing your somatic cell nuclear transfer, we're not implanting the embryo. We're not doing reproductive cloning, but we're making embryonic stem cells to make tissue for you with your own DNA. And we'll talk about this more in regenerative medicine. This is just another idea so you can understand what therapeutic cloning is. So using your DNA to make new tissue for you. Like I said, we'll talk about this with regenerative medicine in the next lecture, but there is a lot of stem cell trials going on. So you can click on this link and it takes you to the clinicaltrials.gov, and you can see what kind of stem cell research people are doing to the point where they're trying it. We'll see if this loads with humans. I like this stem bucks just because I'm sure it's super expensive. Right, so you can see here they're using umbilical cord stem cells to treat diabetes and a lung. They're using human mesenchymal, so these are your adult stem cells, bone marrow, treating optic nerve issues, Crohn's disease, hormone deficiencies. So lots of work being done to use stem cells. There's almost 2,000 studies going on, so kind of cool to look at. Lots of work on stem cell treatment, so regenerating tissue. This is a cardiac tissue regeneration. So you're going to be looking at some of the stem cell therapy in your assignment. So you've been assigned to a group. You're going to look at stem cell therapy for that. So I can't remember what they all are. So I don't have my stuff with me. I know one's diabetes, I think. Sorry, let's see. Why don't I just... No, don't end the show. Okay, we'll look at it in a minute, I think. Anyways, this is a pretty cool TED Talk about regenerating cardiac tissue, if you're interested. This one was really cool, I thought, regenerating the cornea to a healthy eye. It was done on 252 patients with a 75% success rate. And here's some working on joints. Okay, let's see. So, lots of cool stem cell research going on. There's also stem cell treatments that are commercially available. And so that's the other part of your assignment, is to go research one of these. And regenerative medicine ties into what our next topic will be. But it's kind of interesting. So you can get a stem cell facelift. And a lot of these commercial are using adipose tissue, so your fat tissues. This is trying to treat arthritis. There are some I call dubious stem cell clinics that you can look up. So there are places in Colorado that you can find that do stem cell treatment, and that's going to be part of your assignment to look up, and we'll discuss it in class. Here's a Colorado one you can look at. Talks about the procedure. They're taking your bone marrow. They're separating out the stem cells and then injecting the stem cells back into your body for natural healing. So it's pretty exciting. Just have to see how well it works and see what you guys come up with. Couple terms I want to make sure you understand. So if they talk about allogenic, allo means same. So this is a matched donor. Okay, so this is a donor giving stem cells. Auto means yourself. So autologous sources means from you. So they've taken your stem cells and giving them back to you, right? So allogenic is how we do organ transplants right now, right? You have to have a matched donor to get a liver transplant or a kidney transplant. The idea with autologous is we can maybe get stem cells and create your own tissue. If they talk about homologous use, that means you're using the same type of tissue. Whoa, not watching what I'm doing. Same type of tissue for the stem cells and the treatment. Right, so if they were harvesting your bone marrow, growing up more blood cells in the lab and then giving it back to you, that would be homologous. Most of this type we're talking about is non-homologous. It means the stem cells are from different tissue, different tissue source than treatment tissue. Okay, so this is like taking fat stem cells and putting them into your joints, or taking those fat adipose stem cells and doing a facelift with them. So just a couple terms to make sure you are clear on. Lots of potential uses for stem cells, right? Treating disease, wound healing. Wouldn't it be amazing to do spinal cord injury, Crohn's disease, cancer, muscular dystrophy, diabetes? So we're going to be talking about some of these diseases and how they're trying to use stem cells. So, in summary, the goal of the stem cells that we're talking about is kind of basically for fixing your tissue. We've talked about some different ways that you can generate your own personal stem cells, right? Induced pluripotent, embryonic, I guess we didn't do this, SCNT to make embryonic stem cells, blastocysts from in vitro fertilization, using those embryos to make embryonic stem cells. Using your own, right? Using your own DNA is going to be the best way for reducing the chance of rejection. But we're still trying to figure out how to do this. So I hope that gave you a little bit of information into stem cells, and I look forward to discussing some of the applications with you. Thanks.